Very nice to be back with everyone. And I guess this is one of the more informed audiences I've ever given this lecture to. So I'm going to start out with a couple of words about the epistemology of oncology, epistemology of the study of how we know what we know. And oncologic knowledge is largely derived from clinical studies, and that knowledge assumes integrity and freedom from bias. And the challenges are these assumptions true? And if they are not always true, what are the implications regarding therapeutic knowledge? Bias in research refers to errors in design, implementation, or analysis that deviate results from true findings. And these commonly lead to either an overestimation of true benefits or an underestimation of harm. And research integrity is about the steps that we take to minimize bias. Bias is important for several reasons. Scientifically, it leads to the potential for scientific harm due to compromised generalizability of the data, direct harm to patients by causing misleading expectations, societal harms when the data is used for inappropriate resource allocation, and finally, reputational harm to oncologists and to the oncology as a profession due to loss of credibility. And these are what I refer to as the perils of bias. The term scientific skepticism encourages alertness to potential for bias in research, and this is an important part of professional integrity. Scientific skepticism should not be confused with nihilism, and it requires critical evaluation skills, and some of which we're going to discuss today. And a wonderful aphorism that I quote all the time to my students is that not all skeptics are scientists, but all good scientists should be skeptics. So in evaluating research data, we have two approaches. We have the approach of looking at data through rose-colored glasses with great enthusiasm of the wonderful breakthroughs, or using an approach of scientific skepticism and asking the questions, are these benefits really real? Sources of bias can derive from design issues, and I'll just walk through some of these. When there's a no control arm or a substandard control arm, from surrogates because of the limited predictive reliability of surrogates. In non-inferiority studies, when you've got a high non-inferiority threshold, that can give a false impression of non-inferiority, which is actually not true. And there are multiple issues of crossover which can impact on post-progression survival or post-progression treatment and impact on overall survival. Early stopping rules as well can influence the interpretation of survival benefit. In terms of study implementation issues not intrinsic to the design, but in the implementation, where you have substandard post-protocol treatment, this can distort overall survival. We have a common problem in terms of quality of life, where quality of life data is collected, but there is publication bias in reporting of quality of life data, and often very, very delayed reporting. And issues that it can affect analysis, things like informative censoring can substantially impact on the analysis of surrogate outcomes. And we're going to talk about conjectural findings from exploratory and unplanned analyses of either the surrogate outcome findings or overall survival findings. So this is a map, essentially, of the multiple different sources of bias that can be introduced into clinical studies, and this is all covered in a study that in a paper that the ESMO Magnitude of Clinical Benefit Working Group published in 2001, and this article will be made available to you. And most of the information in this talk is actually derived from that paper. So essentially, based on this analysis, and one can develop a checklist of design issues, implementation issues, and analysis issues to check for when reading papers. And what we're going to do is we're going to walk through some of the common issues that we find are commonly causing bias. So the issues we're going to talk about are going to be related to conclusions based on surrogate endpoints. And we're going to talk about predictive reliability, single-arm surrogate studies, and the problem of informative censoring and how they impact on surrogate endpoints. We're going to talk about factors that can distort overall survival data, particularly this issue of lack of what's called appropriate crossover and substandard post-progression treatments. And then I'm going to talk finally about some of the analytic issues with conclusions based on conjectural rather than confirmatory findings. So let's leap into surrogate endpoints. The first key message is that all surrogate endpoints have limited predictive reliability for true clinical benefit. The validity of a surrogate depends on its predictive reliability of true clinical benefit. Does it predict for either living longer or living better? And the limitation of surrogate endpoints is that they have this variable predictive reliability. No surrogate has absolute surrogacy for true clinical benefit across all diseases and treatments. And the surrogates themselves may or may not directly benefit the patient. Now, there is a hierarchy of surrogates. At the peak, at the top of the hierarchy is disease-free survival, and this is used in adjuvant studies and studies with curative intent. This has moderate predictive reliability for overall survival, and it is claimed particularly by patient advocacy groups that the delay in recurrence in itself, even if it's not predictive of overall survival, that in itself is a benefit for patients because this is time without treatment or disease. Next down the list is progression-free survival, so this progression-free survival is applied in a non-curative setting. The predictive reliability is intermediate and very variable. PFS is not a reliable predictor of either improved quality of life or even delayed deterioration in quality of life. And the PFS itself, just the fact that X-ray images change earlier or later, does not in itself provide direct patient benefit. At the bottom of the list are response rate and in a curative setting, pathological complete response rate. These are weak indicators of overall survival. Response rate is a very weak predictor of quality of life, and it has no intrinsic benefit in and of itself. So when we have a study where the primary endpoint is a surrogate outcome, this is the importance of follow-up for overall survival data, and also, particularly in the non-curative setting, has quality of life been evaluated as a secondary outpoint? Because these can give some degree of verification of the surrogacy when possible, and sometimes when they're negative, they're actually giving you the message that surrogacy has been disproven. So quality of life becomes a very important outcome in surrogacy trials. As I said, it can verify surrogacy for true clinical benefit by demonstrating either an improvement or delayed deterioration in quality of life. The issues arise, however, related to the quality of life data. And ESMO has recently published standards for evaluating published quality of life data. Essentially, we will only credit quality of life data when it has been a secondary outcome, not an exploratory outcome. and it's been evaluated with a validated scale. In the methods, we wanted to see there's a predefined hypothesis and a predefined clinically meaningful threshold. You want to see there's a high compliance, and ultimately, in the results, you want to see that there is a both statistically significant and clinically meaningful significant difference in global quality of life, not in just sub-parameters. And this is a checklist which we've developed, which is available on the ESMO website. The second issue in surrogacy relates to single-arm studies, and single-arm studies using an outcome of overall response rate and disease and duration of response. And what you can see is that looking at the evidence presented to HTA bodies, we're seeing a rapid rise in the number of phase 1 and 2 studies being presented. And in 2020, it exceeded the number of studies being brought to approval on the basis of randomized studies. So are single-arm studies always justified? According to the FDA, they write that they're acceptable for accelerated approval in settings when there's no available therapy and where major tumor regressions can be presumed to be attributed to the tested drug. They're increasingly used when standard treatments don't exist, when standard treatments are clearly inferior to new treatments, when diseases are rare, and in many situations where it is perceived that randomized studies are not perceived as feasible. However, they provide a much less reliable level of evidence for real clinical benefit, so it leaves us with a much greater degree of uncertainty. Often, there has actually been a reasonable alternative therapy that could have been used as a comparator, which they have not used. And the use is not always based on empiric evidence of the infeasibility of randomized studies. We know that the overall response rates observed in single-arm studies are often inflated, because when you take the same drug into a randomized clinical study, on average, the response rate is about 8% lower in the randomized study. And in fact, you know, this is an illustration of the problem. These are in yellow these are all drugs that were approved on the basis, or many of these drugs that are withdrawn were approved on the basis of these single-arm studies. So the data is weak, and when they do confirmatory studies, the confirmatory studies do not confirm the degree of benefit. In 2021, this is a very nice study which evaluated 31 approvals, FDA approvals that were based on single-arm studies, and they found that an alternative treatment could have been used as a control arm in 28 of the 31 studies. In 5 of the drugs were approved despite having inferior efficacy to drugs that were previously the standard of care. In more than 85% of them, it would have been feasible to conduct a complete randomized study within a reasonable duration of time. And many of the approvals were not for rare conditions, but they were for common conditions. And this study concluded that this was not good science. It often generates wasteful accelerated approvals that are subsequently rescinded. And many of these accelerated approvals linger without having the confirmatory studies done. Okay, the last issue that I'm going to talk about related to progression to surrogate endpoints is the issue of informative censoring and the impact of informative censoring on surrogate outcomes. And I'm very grateful to Ian Tannock, because what I'm going to be saying is based upon his work. Censoring refers to patients who don't complete the study in full and drop out without further measurements. When there are balanced dropouts between the two arms of a comparative superiority study, it's assumed that this is not going to impact on the results. This is called uninformative censoring. However, and in contrast, when the dropouts, when there are more dropouts in the treatment arm or the experimental arm, due to either a lack of effect or side effects, this can lead to unbalanced dropouts, and this can impact on results. And this is what's called informative censoring. Because patients, and particularly in PFS studies, patients who drop out before progressing for reasons other than deaths are no longer being evaluated. So when you have more censoring of patients on the experimental arm than in the control arm, and you're censoring these poorly performing patients, this can lead to exaggerated progression-free survival benefit. And this is a classic illustration. This is a study of everolimus in his first line, along with hormonal therapy, in first-line postmenopausal estrogen receptor positive breast cancer patients. And in this study, they found a very large difference in PFS. There was a lot of informative censoring. 24% of patients on the everolimus arm dropped out because of toxicity, compared to 6% on the control arm. And ultimately, when you look at the overall survival, there was no difference. This has been a particularly prevalent problem in the whole recent generation of studies looking at PARP inhibitors as maintenance therapy for patients in ovarian cancer. PARP inhibitors have lots of side effects. All of these studies to date have been looking at progression-free survival as the primary outcome. And you can see very, very unbalanced censoring. With almost 20% of patients in this study in the niraparib arm dropping out and thus dropping out of this evaluation. The next issues I want to talk about are the ones that can impact on overall survival data. And we're going to talk about crossover and substandard post-progression treatment. Okay, so firstly, the crossover issue. In studies that are upstreaming studies, are taking an agent which is already approved as a later line therapy, and you're upstreaming it in patients in the control arm, when they progress, should always be receiving the experimental agent, because this is part of the subsequent standard of care. Failure to do this is a massive problem, which is almost endemic. The scientific concerns is that if there is inadequate crossover of the control arm patients when they progress, that means they are receiving suboptimal post-progression therapy, and this can clearly exaggerate the observed overall survival benefits. The second problem is that if the patients haven't been crossed over, the control arm patients haven't been crossed over, it fails to answer the question whether using the same drug is better in the first or the second line. Ethically, the failure to incorporate crossover to ensure standard of care causes harm to the control arm patients. This is an older example. This is looking at the study of abiraterone and prednisone in metastatic castrate-sensitive prostate cancer. So this is upstreaming of abiraterone, which was already approved in castrate-resistant patients. And this was what they found. They found this very substantial improvement of overall survival. This was the data on castrate-resistant patients, and that had shown that had previously shown that abiraterone improved overall survival. And then, so going back to the study in question, what we found was when you look at subsequent treatments in patients in the control arm, there was a lack of crossover to abiraterone in 89%, or to even a biological equivalent in 73%. So only 27% of patients received one of these agents in second-line therapy. So they didn't get standard of care, and this may have exaggerated the benefit. And so we're still left with a question. In patients with metastatic prostate cancer, we don't know if abiraterone needs to be given in the castrate-sensitive or the castrate-resistant setting. In more recent studies, this is the whole spectrum of studies, a range of studies looking at the upstreaming of immunotherapy in renal cell cancer. Immunotherapy has an established role as second-line therapy. These were studies which will bring upstreaming immunotherapy to the first line, and looking at the subsequent therapies in patients in the control arm. And you can see in none of these studies, did more than 50% of the patients receive post-progression immunotherapy. And so, all of these studies showed improvements in overall survival. And still, because of the failure to incorporate appropriate crossover, we don't know if immunotherapy needs to be given up front, or if patients would or would not receive the same degree of benefit if they receive the immunotherapy as a second-line therapy. In contrast to appropriate crossover, when you're upstreaming a study which is a drug which is previously has an established proven role, in a novel agent which is upstreamed, it has no role. It has no established role in post-progression therapy. And so for control arm patients, when they progress, they do not need to be receiving the experimental agent. If you give the experimental agent, one, scientifically, it undermines the ability to determine if overall survival is improved or not. And secondly, there are ethical concerns, because if the crossover delays the initiation of a proven therapy, you're harming patients. And since the new medicine's efficacy is unknown, there is no ethical mandate for the control arm patients to receive the medicine upon relapse. In this same area of patients who've progressed on a surrogate and the impact on overall survival, what happens here in the post-progression phase becomes very important. Most randomized studies involve evaluating a single period of randomization between a novel treatment and an active control, generally looking at a surrogate, with first data looking at surrogates. In studies of first or second line therapies, almost all patients will receive one or more lines of post-progression therapy, which can also influence overall survival. And in fact, in hormone receptor positive breast cancer, it is not uncommon for patients to receive more than 5 subsequent lines of therapy. And I want you to remember that number. So I'm going to show you a couple of different scenarios. This is a scenario 1, where you have an experimental treatment which has an improved progression-free survival. All patients receive optimal post-progression therapy, and the benefit persists. And a good example of this was what was observed when trastuzumab was first tested in metastatic breast cancer. An alternative scenario is where there is a big benefit in PFS, but after optimal post-progression therapy, that benefit disappears or becomes trivial. And this was what was seen when bevacizumab was used in first-line metastatic breast cancer. And this data subsequently led to the withdrawal by the FDA of the approval for bevacizumab. The problem arises when patients have not got optimal post-progression therapy, because it may be that the lack of the suboptimal post-progression therapy may have exaggerated the impact of PFS gain on the ultimate overall survival. And this underscores the importance of credentialing centers to make sure they have the capacity to provide the standard of care post-progression therapy, and also documenting post-progression subsequent therapies until death. And in fact, in the IHC guidelines, they say very clearly, efforts should be made to collect all data pertinent to relevant outcomes, including the occurrence and timing of intercurrent events. Clinical trials are less generalizable if the sponsor tries to avoid or minimize these issues, and post-progression treatments constitute an intercurrent event that is pertinent to overall survival. Now, let's look at a great example, not a great, it's a scary example of a problem. This is MONALEESA 7, a study looking at the role of the addition of ribociclib to endocrine therapy in premenopausal patients. So these are young, fit, premenopausal women, first-line therapy with or without ribociclib. And this study showed a substantial improvement in progression-free survival, and subsequently also in follow-up, an improvement in overall survival. Wherein lies the problem? The problem lies here, that this is the number of patients who received any subsequent treatment. In the placebo arm, it's 27% of the patients received no further subsequent treatment. And it is plausible that the failure to provide subsequent standard therapy to this 25% of patients who progressed on the control arm may have exaggerated the overall survival gain from ribociclib compared to placebo. And this becomes even more intriguing when you look at the forest plot. Because looking at the forest plot, this is a drug that demonstrated no significant benefit in either North America or in Europe and Australia. All of the benefit that was observed was derived from this Asian cohort. So it begs the question, did these Asian patients receive suboptimal post-progression treatment? And it is plausible that the overall survival gain may have been much smaller had patients received the standard treatment. Now, I contacted the principal investigator to raise this issue, and he tried to get the data from Novartis, and Novartis would not provide the data on a breakdown of subsequent therapies, either to the principal investigator or to me. So this is also a terrible issue of lack of data transparency. The last issue that I want to address is conclusions that are based on conjectural findings. And I want to make this distinction between confirmatory and conjectural findings. Confirmatory findings are derived from the primary or secondary analysis of the study, and they may include pre-specified subgroup analyses where there is appropriate adjustment for multiplicity testing and alpha splitting. And one also needs to have a justified statistical plan and ultimately a statistically significant positive outcome. A conjecture, per definition, is an unproven proposition suspected to be true based on preliminary supporting evidence. And in clinical studies, conjectural findings relate to findings relate to the evaluation of efficacy based on incomplete or suboptimal data. And these include findings from either exploratory endpoints or subgroup or exploratory analyses that were outside of the statistical plan. So just to tabulate this distinction, the analysis these are confirmatory outcomes are primary or secondary endpoints, conjectural are post hoc or exploratory. In the confirmatory outcomes, the subgroups are pre-specified, the statistical plan is pre-specified. Conjectural, these are post hoc analyses, exploratory analyses that didn't have a pre-specified statistical plan. there's often no adjustment for multiplicity. So conjectural findings have a higher risk of false positive or false negative findings. And this is why it's important to make this distinction. Now, when is it common to see these? It's common to see post hoc conjectural analysis either when, in situations where the primary analysis showed a very small benefit, and the post hoc interest is to identify subgroups who are more or less likely to derive benefit, which is a very reasonable question. And the other common situation is when the primary analysis shows no significant benefit, and there's a post hoc interest in identifying, well, was there a subgroup where the patient, where the treatment may have been effective? And different organizations have different guidelines. The IHC warns that extort the findings from post hoc subgroup analysis should be interpreted cautiously. The FDA, interestingly, has a draft guideline which has not been ratified and expresses concern that investigators or sponsors' incentives can influence the choice of analyses to identify one or more positive findings. And the EMA has a structured approach saying that they will consider evidence based on conjecture where the subgroup of interest is well defined and clinically relevant, when it's biologically plausible, when there are substantially different results, and where there's been replication of similar subgroup findings from other relevant studies. So here is a great example. This is one where the primary analysis showed a very, very small benefit. This is the addition of pertuzumab to trastuzumab in HER2 overexpressed breast cancer in an adjuvant setting. This was the primary analysis of the invasive disease-free survival rate, and you can almost get a laser pointer between the two curves, almost. In the protocol, they write that exploratory analyses will be performed for DFS to ascertain whether the magnitude of effectiveness might differ according to patient subgroups, and they looked at 12 different subgroups as post hoc analyses. And this is their forest plot, and then they highlighted this issue of nodal status. Now, if you look here, the p-value for the interaction is not significant, but then they still mapped out this curve, and they said, aha, we have found an improvement in disease-free survival of about 2.5% in patients who are node positive. So, and this became the basis for the FDA and EMA approval. So the concerns are, one, that this is one of 12 exploratory subgroups. The interaction was not significant, and there was no adjustment for multiplicity. Many countries are using this, and it's reimbursed. As I believe Canada looked at this data more skeptically and did not approve it. And this is an example where the primary analysis showed no benefit. This is the atezolizumab added to nab-paclitaxel in triple negative breast cancer as a first-line treatment for metastatic disease. This had hierarchical testing for overall survival, first in the intention to treat population, and only then in the PD-L1 positive population. And in this study, looking at progression-free survival, there was a small benefit which became greater in the PD-L1 positive subgroup, but in the overall survival, the overall survival was not significant, and yet they still looked at the overall survival data and got an accelerated approval based on that data, which again was subsequently rescinded. So what I have tried to highlight here is the difference between the rose-colored glasses approach of just looking at the data and regurgitating it uncritically, and using a structured approach to critically evaluate the data to see, to look for evidence, for evidence of bias. Coming back to the question I started with about the epistemology of oncology, about the assumption, I asked me questions. Are the assumptions of integrity, freedom, and bias true? The answer is that in many cases they're probably not. And in terms of the implications for therapeutic knowledge, we need more humility and less hubris. And integrity demands honest disclosure and a deeper evaluation and routine evaluation of these potential sources for bias. When it comes to the role of HTA bodies, all of these issues that I've highlighted make it very difficult for HTA bodies to evaluate if there's real added benefit to justify the expenditure of limited healthcare resources. And this then becomes not only a clinical challenge and a scientific challenge, but also a public policy challenge. So just to conclude again with this wonderful aphorism, not all skeptics are scientists, but all good scientists should be skeptics. What I wanted to do today is to review some of the key tools and the key things to look for in reading clinical research with an appropriately critical eye and a sensitivity for bias. Thank you very much.
Nathan, thank you so much. So I invite everyone to turn on their cameras, and please raise your hand if you have a question for Nathan to kick off the discussion.
I can kick off the discussion.
Go ahead, Ian. Yes.
I mean, that was great, but depressing, Nathan. But I mean, we've just been writing a couple of commentaries about recent adjuvant trials that are making just huge amounts of money for companies. So there's the adjuvant pembrolizumab trial for renal cancer, Keynote 546, and the monarchy trial for abemaciclib. And both of these trials are just fatally flawed, but the companies are managing to push hugely expensive drugs into earlier stages of disease. So 2 years of adjuvant abemaciclib in the United States costs more than $300,000. And yet you pointed out the ways they do this. There's informative censoring, which they don't even recognize or talk about. And, one of the ways that the companies do it, as you pointed out on one of your slides, is they do these global studies. So these are huge studies, and they get people from all over the world to do them. But of course, as you point out, the people in these middle-income or lower-income countries can't afford the drugs. So in monarchy, which we're just working on a paper now with your colleague Bishal and others, basically, the biggest accruers to monarchy were in Brazil, in Mexico, and in China. And you just know the women in those countries are not going to be able to purchase the abemaciclib or another light drug. And so the trials are purposely, in my opinion, biased from the very beginning by the companies.
All right, so this is this issue of credentialing of centers. And in fact, it is a consort requirement as well that the centers should be accredited to be able to provide the standard of care, I agree. I believe, like you, that this is a global strategy. On the issue of informative censoring, it is different it is often different in DFS studies from progression-free survival studies, and in fact, we actually had a journal club this morning exactly on this study, and the DFS was actually ITT, intention to treat. So it was not impacted by informative censoring. But we've seen in many situations, this study showed I think at 60 months, a 5-year improvement in invasive disease-free survival, that does not always translate into improved overall survival. And time will tell. And in the meantime, there's this pressure on countries and on political systems, on healthcare systems, to pay this very extravagant sum. And the question is, why 3 years? Why not 1 year? And the answer is because 3 years, they will make more money off 3 years.
Absolutely.
David, please go ahead.
Yes, I've given something like 70 odd talks to skeptics in the pub, which I enjoyed it while I could still get around. But the trouble is, the term skeptic has now been adopted by the anti-science brigade, the vaccine deniers, you name it, whatever sort of scoundrel you want to name, they call themselves skeptics.
Yes.
And this is awful. I removed skeptics from my Twitter biography.
Well, I said that not all skeptics are scientists.
Right, yes. You can say that again. Do you think it's getting worse, though, in the age of Trump?
No, I mean, I think my problem is that people read studies with rose-colored with optimism bias.
Yes.
Yeah. People would like to feel that they are more clinically empowered than they actually are, and I think that there is a lack of scientific skepticism in reading, because people want to think, they want to feel that they've got really good arrows in their to throw at cancers, and in fact, I'm the constant party pooper in our journal clubs. Because I constantly find myself in the role of trying to watering down the enthusiasm, and it is not contagious, unfortunately. There's a cultural issue as to whether one is going to look at data optimistically or look at it critically, and many people just do not have that culture and do not want to adopt the culture, and don't see the reason for adopting culture of being critical readers.
Yes, no, I agree entirely, and I think I've been a bit of a party pooper. Just the other day, Folcadine was banned from cough suppressants, so I was busy saying, "Don't worry, it didn't work anyway," which it doesn't, of course. And there were pharmacists on the radio saying, "We can recommend something else," which also doesn't work, of course, for coughs. Oh dear. Thank you very much.
Great pleasure.
It's a terrific talk.
Indeed, I have a follow-on question, and maybe it's for both Nathan and Ian, so you decide. We can contribute both. So Nathan, you presented very important examples of trials, which is wonderful teaching material, wonderful ways to understand when and how things go wrong. If we take, let's just focus on oncology. If we just take oncology drug approvals over 10 years,
yes,
what proportion of these studies do you think, registrational studies, have been depicted in such a way, in the public way, so where we can learn from them, and how much more do we have left to do? Is it makes sense to look back?
Well, I was asked by Nature Clinical Reviews to look at all of the drugs approved by the FDA over the past 5 years. And what I found was that 70% of them were approved on the basis of data just from surrogate data. Only 30% of them had overall survival data, and even those that had overall survival data, the problem of inadequate crossover was widely prevalent. Mm-hmm. So these are big issues. When we applied the ESMO MCBS to these 160 approvals, we found that only about a third scored highly, a third scored very low, and a third was somewhere in the middle. And so even those high ones may not be as good as they really are. in many cases.
Indeed, because when you start depicting, there are so many other parameters which might not meet the criteria. Thank you. There's one question in the chat from Sophie, which I will read, and then I'll pass it on to Lydia. So what do you think of yesterday's announcement by Jeremy Hunt that the UK MHRA will speed up the approval of drugs based on the presentation? How concerning is this?
Again, who's going to speed up the approval of drugs?
The British decision maker, the British regulator, because now it's not the EMA, it's now MHRA
Right.
that decides.
Okay. I mean, the speed of the approvals is a reflection of efficiency. The quality of the approvals is a more important important decision. The policy of the FDA has been, has been to enhance inclusiveness, to lower the threshold. So is there anything that has a chance of being beneficial to approve, and to lower and to have a very low level of discernment. And what will be the policy of the English licensing authority? Now it's not so matter of issues of speed as what will be their thresholds.
Mm-hmm. Yes, very interesting space to watch. EMA is also under a lot of pressure and discussions, speeding things up. So, Lydia, please go ahead.
Yeah, I have a very general question. As you know, we are supporting investigator-driven trials, non-commercial trials, and from interaction with investigators, we learned the hard reality that if you want to collect the overall survival data, then you have you need to have your study open like for 10 years, much longer periods, which makes it tremendously expensive if you're in the non-profit or public supported trials. So some people are suggesting that maybe if from the start on, you could do what they call the registry-based RCT, where you could close your study with a surrogate endpoint, but then follow up with e-health records or the administrative databases. Could that be something acceptable if it's, of course, if it's pre-specified, or do you have other solutions to lower the cost of collecting overall survival data?
Particularly in the post-progression setting, the issue is the completeness of the data. So, e-health records, one should be able to get detailed accounting for post-progression therapies, and which is going to take work to do it, but the information should be readily available, and also the time of death is going to be available. But it still requires a commitment of manpower to derive this data from the e-health records, and because when it's not done, you can see what happens. If you don't know what patients have received from the time that they progressed until you've got no idea if they've received standard of care or not, or if they've received popcorn and chocolate milk. And that is a problem, and the quality of data collection about subsequent therapies, even in sponsored studies, is an issue.
Thank you.
Thank you, Nathan. Any other questions?
Another problem, I think, for the UK government is that they identified as a growth area nutraceuticals, and there is no area where the evidence is worse than for the supplement industry. It's another aspect of Singapore on Thames, I'm afraid.
Yeah.
Okay, I have another question, Nathan. So you mentioned in your presentation, and you put examples where justice happens sometimes, and we withdraw drugs from the market which went through accelerated approval on single arm trial data, and but we know that this withdrawal happens years after, and during this time the company manages to make a really large sums of money.
Yeah.
And what do you think we can do here? Can there be any legislation around it to have the company liable to return this money to providers, to patients? Because that's one thing is just the earnings on something, on the hype that doesn't work. And another thing is potential harm of patients being on a toxic drug or just being on that drug
But
instead of, let's say, not being treated.
this is the role of a strong HTA body. We've been studying HTA decision making. We had a project running with the London School of Economics, and we know that when a drug is approved on a single arm study with an accelerated approval, firstly, HTA bodies deliberate a lot longer. So even though the approval has been accelerated, the HTA process is slowed down. And we also know that in the majority of cases, there will also be some form of negotiation on price, and to give these drugs market access. So the price that is ultimately being paid for by governments is generally substantially lower than the US base price, which is a point for negotiation. But it would also be totally reasonable to say that until the price will be much lower, as long as there is a contingent or an accelerated approval rather than a full approval. I don't think whether one can negotiate a payback. I don't know if anyone. I've never heard of an HTA body trying to negotiate that.
That why it was my question, because
Yeah.
I think an HTA body will tell you it is not their remit and this is a political decision.
Yeah.
And that's one point. And if we look at the proportion of earnings, what is coming from the US market for each drug during that period versus market
it is very high. It is
very high. So
and you see, the other thing is that because of the idiosyncrasies of the Medicare Part D laws, such that the Medicare is the largest single purchaser of drugs in the United States, and Medicare must provide all FDA-approved drugs at a price set by the companies without price negotiation. So there's no relation. So with this suspension of market forces, there is no relationship between the clinical value of the drug and the cost of the drug. That's
very, very important words. So Ian, comments, questions from you?
Yeah, I think it comes down to a matter of the people who are sitting on the guidelines committees and sitting on the FDA and the EMA advisory committees. Unfortunately, we already know that, if you're invited to go on one of these committees, the amount that you declare under the Sunshine Act goes up. And so we don't have, we need to be unbiased, and yet we don't appoint committees who are even financially unbiased. And I think the only way for the future is we have to do a better job of educating people who, oncologists who sit on these committees. But we need to have committee members who can't make profit out of what their decisions can lead to. And I don't know, the counter argument, well, if we did that, we'd never get anybody to serve on the committee. Well, there are some of us who don't take money from companies. And I just think that has to become more and more acceptable. They have tightened up. Universities have tightened up, and I think the whole system needs to be tightened up.
both the EMA and the FDA, their mandate is to approve drugs that are safe and effective. And so their threshold for defining effectiveness is anything which is either equivalent or better than the pre-existing standard, and it doesn't need to be better by much. It can be trivial. But approval does not mean market access, and this is why HTA bodies are so important.
That's true.
Absolutely, but they also have a very strict remit what they can and cannot do, and there is a big political reason behind that. So I really think there is a need for another layer here, either give more power to the HTA bodies, or have another layer of legislation or the government playing the role. And there is one comment in the chat saying that there will be pricing negotiation in Medicare in the next few years. It has already started. And
very limited, with a very small number
extremely
of drugs.
limited, and unfortunately, it has nothing to do with newly approved drugs. This is only for drugs who have been on patent for at least 7 to 8 years, as I understand. So everything we're discussing right now, Medicare negotiation, will not resolve in any way. Lydia, final comment question from you as we wrap up.
I just want to say that with the revision of the pharmaceutical legislation in Europe, we are now pushing hard for kind of mandatory scientific advice for the design of the clinical trials, for the registration trial, with input from HTA. So that at least the design of the trial, the endpoints are kind of approved by HTA, but that should be the joint HTA. Then in Europe, we still have that complexity. Do you believe that that's an approach that could help?
I think in randomized clinical studies, particularly with upstreaming studies, if the Helsinki committees, the IRBs, were first forced to look at this issue of crossover and credentialing as part of the approval process for a study and for a site approval, I think that would make a substantial step forward. I've been trying to push this with the national Helsinki committee or IRB committee, looking at randomized studies, and I think and they are open to listening to this. So let's just say, particularly with expensive drugs that are being upstreamed, that the sponsor would need to ensure that the patients on the control arm will have access to the drug post-progression as part of their post-progression therapy.
Great.
Lydia, just one of the comments from experience of scientific advice. We need to remember that this advice is optional for companies to take on board, and they need to see what the regulator suggested, and mostly the FDA. And if the US is the key market, and that study is faster and easier to do, not considering what UNETA suggested, chances that they will take those recommendations on board are slim.
Yeah.
So plus, let's not forget the advice is highly confidential, and we will never find out what has been told to the company and what has happened in the end. So that's another massive lack of transparency, and for us to check potential compliance with good practice. So